Protein biosynthesis is the process whereby biological cells generate new proteins. It is balanced by the loss of cellular proteins via degradation, or export.Translation. The assembly of amino acids by ribosomes is an essential part of the biosynthetic pathway, along with generation of messenger RNA, aminoacylation of transfer RNA, co-translational transport, and post-translational modification. Protein biosynthesis is strictly regulated at multiple steps. They are principally during transcription and translation. The cistron DNA is transcribed into the first of a series of RNA intermediates. The last version is used as a template in synthesis of a polypeptide chain. Protein will often be synthesized directly from genes by translating mRNA. However, when a protein must be available on short notice or in large quantities, a protein precursor is produced. A proprotein is an inactive protein containing one or more inhibitory peptides that can be activated when the inhibitory sequence is removed by proteolysis during post-translational modification. A preprotein is a form that contains a signal sequence that specifies its insertion into a through membranes, i.e. targets them for secretion. The signal peptide is cleaved off in the endoplasmic reticulum. Pre-proteins have both sequences still present. In protein synthesis, a succession of tRNA molecules charged with appropriate amino acids are brought together with an mRNA molecule and matched up by base pairing through the anticodons of the tRNA with successive codons of the mRNA. The amino acids are then linked together to extend the growing protein chain, and the TRAS no longer carrying amino acids are released. This whole complex of processes is carried out by the ribosome, formed of two main chains of RNA, called ribosomal RNA, and more than 50 different proteins. The ribosome latches onto the end of an mRNA molecule and moves along it, capturing loaded tRNA molecules and joining together their amino acids to form a new protein chain. Protein biosynthesis, although very similar, is different for prokaryotes and eukaryotes. Transcription. In transcription, an mRNA chain is generated, with one strand of the DNA double helix in the genome as a template. This strand is called the template strand. Transcription can be divided into three stages: initiation, elongation, and termination, each regulated by a large number of proteins such as transcription factors and coactivators that ensure that the correct gene is transcribed. Transcription occurs in the cell nucleus, where the DNA is held. The DNA structure of the cell is made up of two helixes made up of sugar and phosphate held together by hydrogen bonds between the bases of opposite strands. The sugar and the phosphate in each strand are joined together by stronger phosphodiester covalent bonds. The DNA is unzipped by the enzyme helicase, leaving the single nucleotide chain open to be copied. RNA polymerase reads the DNA strand from the 3' end to the 5' end, while it synthesizes a single strand of messenger RNA in the 5' to 3' direction. The general RNA structure is very similar to the DNA structure, but in RNA the nucleotide uracil takes the place that thymine occupies in DNA. The single strand of mRNA leaves the nucleus through nuclear pores and migrates into the cytoplasm. The first product of transcription differs in prokaryotic cells from that of eukaryotic cells, as in prokaryotic cells the product is mRNA, which needs no post-transcriptional modification, whereas in eukaryotic cells, the first product is called primary transcript, that needs post-transcriptional modification to give hnRNA. hnRNA then undergoes splicing of intrasphere spliceosomes to produce the final mRNA. Translation. The synthesis of proteins from RNA is known as translation. In eukaryotes, translation occurs in the cytoplasm, where the ribosomes are located. Ribosomes are made of a small and large subunit that surround the mRNA. In translation, messenger RNA is decoded to produce a specific polypeptide according to the rules specified by the trinucleotide genetic code. This uses an mRNA sequence as a template to guide the synthesis of a chain of amino acids that form a protein. Translation proceeds in 4 phases: activation, initiation, elongation, and termination. In activation, the correct amino acid is joined to the correct transfer RNA. While this is not, in the technical sense, a step in translation, it is required for translation to proceed. The AA is joined by its carboxyl group to the 3' of the tRNA by an ester bond. When the tRNA has an amino acid linked to it, it is termed charged. Initiation involves the small subunit of the ribosome binding to 5' end of mRNA with the help of initiation factors, other proteins that assist the process. Elongation occurs when the next aminoacyl tRNA in line binds to the ribosome along with GTP and an elongation factor. Termination of the polypeptide happens when the A site of the ribosome faces a stop codon. When this happens, no tRNA can recognize it, but releasing factor can recognize nonsense codons and causes the release of the polypeptide chain. The capacity of disabling or inhibiting translation in protein biosynthesis is used by some antibiotics such as anisomycin, cyclohexamide, chloramphenicol, tetracycline, streptomycin, erythromycin, puromycin, etc. Events following protein translation. The events following biosynthesis include post-translational modification and protein folding. During and after synthesis, polypeptide chains often fold to assume so-called native secondary and tertiary structures. This is known as protein folding.